Hello, everyone, and welcome. My name is Manolis Kellis. I'm a professor in the Computer Science and AI Lab at MIT and also a member of the Broad Institute. Today, I'd like to tell you about our work on deciphering human disease at the single cell level. So our goal is to generate massive amounts of data and integrate these data and use them to make an impact on human biology and disease. Our lab integrates systematically large-scale genetic and genomic datasets in order to decipher the mechanism through which these genetic variants are acting. And we do that by profiling both the transcriptome and the epigenome of thousands of healthy and disease samples, using all these data to develop AI and machine learning algorithms that integrate them in order to predict what are the driver genes, regulatory regions, and cell types that underlie complex traits. And then we validate our predictions systematically using high throughput technologies that we have developed in cell cultures, but also in several collaborations using animal models to validate the predictions. And then we disseminate all of the results and start all over again. So we have now generated massive pipelines for single cell dissection of human disease across brain, cancer, metabolism, and multiple other disorders. In brain alone, we're looking at Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's disease, psychosis in the context of AD, schizophrenia, bipolar, Down syndrome, autism, depression, suicide, post-traumatic stress disorder, and aging. Across more than 1,500 post-mortem samples, capturing more than 7 million individual cells from these individuals, and then distinguishing across multiple tissues more than 60 different human cell types, each profiled across hundreds of individuals. The end goal is to understand how this variation at the single cell level affects different regions of the brain, different organs in the body, and different molecular processes across RNA, across epigenomics through DNA accessibility, DNA modifications, histone modifications, and DNA accessibility. And we're using this in the context of multiple tissues, as well as embryonic developmental stages and primary cells across different stages of differentiation to systematically map the human epigenome and understand how it underlies human disease. So we have actually been involved in both the data generation aspects. We coordinated the integrative analysis of the Epigenome Roadmap project, and my own lab has generated one of the largest resources for single cell data in the human genome ever. And we've used this also in numerous collaborations to start understanding the mechanism of human disease. In particular, for Alzheimer's, we've been studying multiple major cell types of the brain and how they change in the context of Alzheimer's. The first lesson is that the major component of variation is actually the different cell types from each other. But as we start looking across early, intermediate, and late stages of Alzheimer's disease, we can see how the phenotypes are changing from high cognition and then low pathology to intermediate pathology and reduced cognition to very low cognition and high pathology. And we can see what are the gene expression changes that are happening at the single cell level for each of those data sets. So every dot here represents a different cell, and the clustering is based on their global gene expression profiles. So what we're noticing here is that within excitatory neurons, we find two major clusters. One of them is associated with Alzheimer's, and the other one is associated with low Alzheimer's disease. And this is true across all of the major cell types. If we subcluster these major cell types completely de novo, what we're finding is that there are subclusters of excitatory, of inhibitory, of astrocytes, of oligodendrocytes, of OPCs and microglia that are all partitioning the disease phenotypes. So individuals who have higher amyloid burden, more advanced stages of cognitive decline, more advanced stages of pathology and AD, these individuals are showing a different subset of cells much more frequently for each of the major cell types, indicating that pathology and cognitive decline are in fact leading to major global changes in the context of the transcriptional signatures of disease. The second thing we're learning is that if you look at the sex differences of these individuals, we're actually finding that female individuals have many more cells from the sicker cell types than male individuals, indicating that women are in fact showing a stronger signature of disease at the transcriptional level and are perhaps more resilient because those individuals were in fact matched between men and women for their pathological scores. We're also exploiting somatic mutations across these data sets to basically understand from the single cell RNA sequencing, can we infer the specific mutations that are happening across each of these RNA molecules and use those as a way to recognize not just common and rare variants associated with disease, but also somatic variants, which are actually happening during development or late in life, given that this is a late onset neurodegenerative disorder. We're also looking at longitudinal cohorts of progression of disease. In this particular example, we're looking at cancer and how one particular patient in a cohort of 100 patients that we're working on in collaboration with Genevieve Bollon over at MGH, we're looking at how these individuals are progressing through disease and how are their genomes actually changing given that somatic mutations are in fact driving a lot of cancer and specifically metastatic melanoma. What we're seeing is that there are convergent mutations across multiple lineages that are coexisting and co-evolving within the same individual, and that those are in fact showing single cell transcriptional changes that are indicative of a lot of immune tumor interactions that are leading to resistance to immunotherapy in some individuals, but response to immunotherapy in other individuals. And we're able to now trace the progression of these changes across the genomic, epigenomic, and transcriptional levels. We're also integrating beyond just one single phenotype, dozens of phenotypes in the context of disease. So we're modeling electronic health records, and we're using those to systematically impute missing information across multiple data modalities in databases of millions of patients. We're integrating doctor notes, billing codes, as well as prescriptions, lab tests, medications, and we're using the multiple data modalities to reinforce each other and complete an otherwise sparse clinical record. We're integrating these data sets in this hierarchical model, as well as genetic information from these individuals to basically start predicting what are the gene expression patterns in multiple tissues of those individuals through the Genotype Tissue Expression Project, the GTEx project, to basically look at what are the tissues and cell types where these gene expression changes are manifesting themselves in coordination with the multi-phenotype clinical records. We're integrating all these data sets to now start predicting what are the driver genes, regions, cells, and pathways underlying human disease. And we've developed these Bayesian causality inference models that allow us to infer the probability with which any one genetic variant is causal based on the global enrichment of all of the genetic variants from that trait in specific epigenomic annotations across our hundreds of cell types, in order to then use this empirical prior with the observed effect size in the genetic association study that implicates multiple co-inherited variants, in order to then predict which ones of those are in fact most likely to disrupt common biological processes and therefore most likely to be drivers of disease. We've developed these statistical models of causality that allow us to now starting with the genetics, predict the intermediate molecular phenotypes at the epigenomic level and the transcriptional level, in order to then understand which ones of those intermediate molecular variables are in fact mediating the effect of genetics on disease. We're systematically building predictors of these intermediate variables across hundreds of individuals, and then using those to break up this very long path between genetics and disease. by imputing systematically these intermediate molecular phenotypes and then using those to start predicting disease in a causality setting. This allows us to now start predicting the driver genetic variants, the specific motifs that they're implicated in, the upstream regulators, and the downstream consequences of those models. In the context of obesity, this has allowed us to now uncover the circuitry that underlies the strongest genetic association with obesity. What we found is that in this FTO obesity locus, which is the strongest genetic association with obesity, we found that the FTO gene itself is in fact not implicated. Instead, what we found is that the single genetic variant alteration inside this FTO region of the genome, which does not perturb the FTO gene directly, is in fact perturbing a motif that is normally bound by the AT-rich interacting domain factor, ARID5B, that binds this AT-rich motif and then represses this very large super enhancer that normally controls genes IRX3 and IRX5. When that regulator, that repressor, can no longer bind, these two genes are de-repressed. And what's really remarkable is that these two genes, in fact, lie 500,000 nucleotides and 1.2 million nucleotides away from the genetic variant that controls them, highlighting the importance of inferring this long-range circuitry in the context of disease. So what do these genes do? These genes, as it turns out, are master regulators of a process known as thermogenesis. This is the process that our fat cells undergo to burn excess calories in our diet and turn them into heat rather than storing them in our white adipocytes as fat. So that can make the difference between lean and obese individuals. Why do we figure out these circuits? Because we can then go and manipulate these circuits. We can now increase or decrease the expression of the upstream regulator. We can increase or decrease the expression of the downstream target genes. We can even go and make single nucleotide alterations that change that single motif in primary adipocytes from risk and non-risk individuals, and show that in each of these cases we can switch between lean and obese phenotypes. We're able to, with a single nucleotide alteration, change the primary adipocytes from being completely unable to generate heat, to thermogenize, into a 7-fold increase back to physiologically normal levels with a single nucleotide alteration out of 3.2 billion nucleotides in the human genome. I mean, that's as precision medicine as it gets. We can then go in and test the effect of IRX3 downregulation in mice. And what we see is that the mice are in fact losing 50% of their body weight when we shut off this pathway through IRX3. Normal mice gain weight, these mice are unable to gain weight. So that gives us hope for the ability to go in through this knowledge and intervene to change disease circuitry. We are now scaling up these studies to change hundreds of constructs at a time. We've developed modular and programmable assays in a series of papers, more recently in collaboration with the CSAIL lab at the Picard Institute, to basically make these manipulations in iPSCs, in induced pluripotent stem cells, and differentiate those into all of the lineages that we care about, in this example, in the brain, and then start systematically dissecting these regions. The last thing is, being in the context of CSAIL, we have now started enabling data sharing in order to increase discoveries. What we found is that there's inflection points in many of these integrative analyses where for decades, sometimes there's not a single hit until cohorts are merged. And even today, the silos that are separating different types of data sets are in fact preventing the integration that I showed you across genetic, epigenomic, transcriptional, phenotypic, and disease variables. The two factors that are driving this, on one hand, are data hoarding due to competition, both in industry and academia, as well as patient privacy, where there are regulatory hurdles to sharing. And the challenge now is that we either have no data or we have full access to the data with no oversight. In collaboration with SAIL, the Secure AI Labs team which is a new venture that I'm a co-founder of out of CSAIL and the Media Lab. We've now developed technologies for federated learning, creating secure enclaves and enabling traceable computation to reward the different parties participating in the discovery accordingly. This allows a full stack, multi-party, secure private computation that enables integration across hospitals, AI, third parties, as well as pharma, each preserving their privacy from their data to the searches that they carry out, and even to the algorithms that are being used. And the data access is being monitored without actually revealing the algorithms and the AI, enabling us to now create these complex ecosystems with multiple parties, being able to all work together and interact as if they all had access to the data without actually revealing their privacy. So that's our work. You can find out more at computationalbiology.mit.edu. My name is Professor Manolis Kellis. This is the basic loop that we're doing, incorporating genetics, large-scale data generation, large-scale data integration, and then systematic validation. So we combine big data and machine learning and large-scale dissemination in order to have impact and to have fun. So thank you for joining our meeting, and we're so excited to interact with you, so excited to tell you more about the work that we're doing, and looking forward to answering any questions you may have. Thank you.